Okay, this is the transformation lab using E. coli and pea green. In this lab, we are performing genetic transformation. We're inserting a gene into the E. coli to change its phenotype, which we're trying to make it glow and grow in the presence of the AMP antibiotic. The stuff we're using to make it glow is also found in jellyfish, and so that's why they glow. The materials we'll be using is a Petri dish, 15 milliliter tubes, pipettes, calcium chloride, inoculating loops, which are like those loops that are like, and then it has a little thing and you can like get bacteria. Laura broth, ampicillin, an ice bucket, Sharpies, a timer, and we'll be using our phones, a 42 Celsius water bath, goggles, glass beads, and an incubator. The independent variable is the plasmids, and so that doesn't depend on the other. And then the dependent variable is Laura broth and E. coli. All right, here we go. So we started off our procedure by washing our hands and putting on some goggles. And we put 250 milliliters of ice cold calcium chloride onto two 15 milliliter tubes, and then we placed both the tubes on ice. So why we place them on ice is to allow the bacteria membrane to stabilize and increase the interaction between the calcium. Then we'll label them positive and negative. The positive tube will contain the plasmid DNA. And then we'll use an inoculating loop to put E. coli on the positive plasma tube, and we'll swirl it around to make sure that it is securely in the tube and all over it. Then we will put the positive tube on ice and repeat step 4 for the other ones, for the negative tube. And then we'll add the negative tube to ice as well. We'll let the tubes sit on the ice for about 12 minutes. And while the tubes are on ice, we'll get our 3 Petri dishes and label them positive, which is the experimental one, and the negative, which is the control. And after 12 minutes is up, we'll take the 2 tubes off the ice and immediately put them into the 42 degrees Celsius water for exactly 90 seconds. So after the 90 seconds is up, we will remove the tubes from the water bath and immediately put them back on ice for another 90 seconds. Then we will use a transfer pipette to add 250 milliliters of Laura broth to both tubes, and we'll swirl them around to make sure that it covers the whole thing and mixes well with the cell suspension. And then we'll place the tubes on a rack for 5 minutes. Then after the 5 minutes is up, we will take out the cells from each tube and spread them on a Petri dish for the positive cell and dish. Then we will use a pipette to add 100 milliliters of cells from the negative plasmid tube to the Petri dish, label negative, and also to the plain one. We will open each Petri dish lid and carefully pour 1 glass bead into each plate. Then we will carefully, we'll shake it back and forth to move the glass bead around so that the entire Petri dish is has been touched by the bead. Then we will need to remove the glass bead, and we will do this by taking the Petri dishes over to a trash can and holding them vertically over it to release the glass bead out of the container. And then we'll use another pipette to add 100 milliliters of cell suspension from the positive plasmid to each positive dish. And then lastly, we will put the Petri dishes in the incubator and let them sit for 24 hours.